welcome to CELLIMA Talks, a webinar series that explores the interface between scientific insight and innovation. In this edition of our format Stories from the Bench, we feature conversations with researchers whose work has shaped the foundations of modern drug discovery. In today's episode, From Screening to Therapy, the long journey of an NK1 antagonist in drug discovery, we feature Mike Snider, a pharmacologist whose work helped lay the foundation for one of the most widely used NK1 receptor antagonists in clinical practice today. With a career spanning academia and industry, including leadership roles at Pfizer and Berlex, Mike reflects on how high-throughput screening, organizational dynamics, and scientific serendipity intersected in the early days of this NK1 program. This is not just a story about a molecule, it's a reflection on how drug discovery really works. Unpredictable, empirical, and deeply human.
Mike, great to see you today. Thanks for taking part in this interview. Let's just step into our webinar now. Could you give us a brief introduction to your background and what you've done in the pharmaceutical industry and what has been your career path over the years?
Sure. You may not know, I'm from California originally, from Southern California, and I'm a product of the 60s, you could say. And California, that was quite an active time culturally. And as a part of that, experimenting with pharmaceutical agents was something that was very common in the late 60s. And I organically just developed a fascination for how drugs act. And I'm not from an academic family. I'm one of the first, actually the first to get an advanced degree of any people in my family. But I learned about pharmacology through a course that I took in college on psychopharmacology, which of course is about drugs acting on the brain and behavior. And I learned that pharmacology was a thing that you could study as an entity. And I applied for some graduate programs and I was accepted at a few and decided to go to Ohio State. And I had no idea, but I was very fortunate that the program at Ohio State was fabulous. It was at a time that was ideal, really, for the merge of classical pharmacology and modern method pharmacology. It was when receptors were starting to be cloned and receptor binding was really reaching magnificent places for understanding how drugs work and how naturally occurring substances in the body work. So it was a great time for learning pharmacology. I'm a big fan of Chauncey Leake, who was a pioneer, really, one of the first huge pioneers in pharmacology. And being at Ohio State recently, being awarded a prize reminded me of Chauncey Leake. He was at Ohio State and attempted to make pharmacology an entity in and of itself by merging the departments of pharmacology and the College of Pharmacy, Medicine, and Veterinary Medicine. It's a fabulous discipline. It relies on biochemistry and physiology. It is not a discipline in and of itself. It uses the tools of physiology and biochemistry in order to understand the drug, rather than to understand the biochemical or physiological system. So I feel fortunate and really blessed to have this background and foundation in science being pharmacology. It's been fun. It's been fun.
Wonderful. Amazing story and very inspiring story. And was this also the reasons or the origin why you was so inspired to pursue a career, especially in drug discovery? And looking back, what kept you mostly motivated throughout your journey? Because you've spent a long time of your life in this discipline.
Yes, that's a very good question. I think I had a young family and I needed to make money. So after postdoc, I postdoced at the Mayo Clinic with Elliot Richardson. My focus was on second messenger eliciting the second messenger responses from bioactive compounds. But the focus of the laboratory was to understand how antidepressants and antipsychotics interact with mostly biogenic amine receptors. So that was fun. And after that, I took an academic position at the University of Michigan. But then the academic or the financial issues started to rise up. So I joined it was a biotech company called T-Cell Sciences. And that was fully into discovery of new entities. And it also served the purpose of my main focus was neuroscience, but my wife is an immunologist, and the year and a half or 2 years I spent at T-Cell Sciences gave me a very good understanding of immunology as well as neuroscience. So that, I think, made me fairly attractive for, at the time, Pfizer, which was building what they called a new leads discovery, which was focused on high-throughput screening. And this was in the mid-'80s, which was when high-throughput screening was really taking off. Subsequent to that, it became really fun to build a new department doing drug discovery research. High-throughput screening is basically boring, but what comes out of it is exciting.
It is exciting. Yeah.
It is fascinatingly exciting, and that's what kept me going. Yeah. The successes is what keeps you going.
Yeah, I completely agree with you. Looking all over the million of data manually to that time was boring, but figuring out the needle in the haystack and getting a clue what could be the mechanisms that are relying on the treatment paradigm you're interested in, that was worth the time you spent it. I would have loved that we had this now available AI tools, machine learning, deep learning, to look all over the data.
Really, drug discovery only starts when you have a good lead compound that's potent. Then the work really starts.
Yeah. Yeah.
Figuring out what it's good for.
Yeah. The fine-tuning and the challenges are occurring. So during your time when you were at Pfizer and later you were also at Berlex, what were the key challenges you faced in early drug discovery, particularly when looking at the small molecules that you have worked with?
Sure. This probably won't come as a surprise, but a high-throughput screening entity within an organization is looked at as a service.
Yeah.
And when there are successes, that becomes a motivator for management to expect successes. So I think the biggest challenge was the pressure, the expectations that you're going to be successful and come up with something that will serve as a starting point for a new lead discovery for a drug. I think that's there. The other challenges, of course, are oftentimes you are presented with targets that are not really amenable to drug discovery.
Mm.
And for instance, this is maybe a little extreme, but we were asked to do it to find an antagonist of an enzyme in the Krebs cycle. What could go wrong there? So it seems like the scientists that are in the actual therapeutic areas sometimes get a little carried away with, gee, wouldn't it be great if we could do X? And we have this high throughput screening group. Let's have them do that. And it's no work for them. We work together with them to put an assay together. At one point, we had a goal, which was more of an expectation, that we would do 12 new screens every year. And it didn't really matter. It was just a numbers game.
Yeah. And as you mentioned, you said it, you worked extensively in high throughput screening, which was fully automated and was looking for miniaturization approaches and also in other innovative technologies. How, in your opinion, did and do these advancements shape the research landscapes during your career?
Really the whole engine of the pharmaceutical business during my career really was based on finding leads using high throughput screening and then putting medicinal chemistry to optimize those leads and assuming that an optimized compound will be useful in the disease it was designed for. That's a big assumption. But that's pretty much shapes up my career in terms of what we did day to day.
You mentioned it already, the NK1 receptor, which I think is one of the highlights of your career.
Yes.
The development of this drug lead now to a medical care system for post-tumor therapy, as far as I recognize it correctly.
Post-op and chemotherapy. Nausea and vomiting are problems after operations and following chemotherapy.
Yeah.
So it's used in both of those paradigms.
How did your lab discover this pharmacophore? And was high-throughput screening prominent in the discovery process?
Yes, it was. At that time, this was in the mid-'80s, right?
Yeah.
This was before we were cloning receptors into whatever cell we wanted to use. We used bovine caudate tissue, had NK1 receptors. At that time, we didn't know that there were species differences between the binding of our compound in different species. That came later. But we found actually a fairly potent compound. It's a quincuridine, basically a heterocycle, 2,3 substituted. Not much different, actually, from what's published in 91, the CP96345. Not much different at all out of the box. I think it was a little under 100 nanomolar as the first discovery. And then putting a 2-methoxy on the 3 position brought it down to single-digit nanomolar. So the actual medicinal chemistry that went into it was fairly quick for that first compound. The quincuridine was changed to piperidine by Manoj Desai, one of the chemists. There was only 2 chemists on the project, but we got rid of the quincuridine and made it into a piperidine with instead of a benzhydryl at the 2 position, just a simple phenyl group at the 2 position. Just a beautiful little simple pharmacophore, highly amenable to. For instance, its absolute stereospecificity in the 2 and 3 position are both chiral. The only compound with activity is the cis orientation, 1 cis orientation. And Manoj Desai worked out a stereospecific synthesis for these compounds, which was quite unique.
And what were the biggest scientific hurdles during the development of this drug, Mike?
Development is different than discovery.
Okay.
Development is, okay, now we have it. Is it safe and is it effective? What is it effective for?
Yeah.
So based on what Pfizer was interested in, we looked in big market diseases, asthma, depression, pain. Ideally, you would think that substance P and the NK1 receptor would be ideally located to modulate neuroinflammation. That seems like its location in the body would be ideally suited for that type of function.
Yeah.
It didn't do anything in pain or inflammation. And I parted ways with Pfizer about the time that they were stuck. Meanwhile, it was a very hot area in the 80s. So the NK1 receptor, substance P, was thought to be a very good target by a lot of different companies.
Yeah.
And fairly soon after the publication, Glaxo made the compound and told us what they were doing. But they had expertise in nausea and vomiting, ondansetron, right? So undoubtedly they ran our compound that they synthesized through their models and found it useful in prevention of nausea and vomiting. I have not talked to anybody from Glaxo, but I'd be fascinated by that story of how they came about that. So they filed a method of use patent for the use of NK1 antagonists in preventing nausea and vomiting. Meanwhile, Merck, who was upset to be mild with our paper in Science in January of 91, they thought it should have been Merck as the discoverer of that. So they put an army of chemists on it to get around the patents. And I'm sure they tried to find other uses for NK1 antagonists as well. I haven't talked to anybody at Merck, but I'd be surprised if they didn't. But it ended up being so far only useful for this one indication. And nobody could have predicted that. Talk about the biggest challenge in development. The therapeutic area guys, they say the expression is up in disease in this place, in this organ, and that means this, or it changes in disease, the expression of this target changes in disease. It may lead to a therapeutic modality, but no guarantees. It's entirely empirical, I think, is the take-home message for development.
Mike, this drug went through various stages and was eventually developed by other companies. What was your reaction when you now learned about its impact, and how does it feel for you to see that the project evolved and ultimately received this recognition and importance?
I was no longer at Pfizer at that time, but I think even if I was there, I would have been thrilled, and I am thrilled by the fact that this baby of ours in this small group at Pfizer actually is treating millions of people. It's useful. Yeah.
So when you look back, what do you think was the most significant scientific insight or breakthrough from this project, even if it didn't lead to the immediate result?
I would go back to my observation that pharmacology is defined by antagonists, and it's entirely empirical. So you're not going to really know what is going to be useful in treating or what condition until you try it.
Okay.
Which is not good news.
That's realistic.
I think it's reality.
That's it, how it is. Otherwise, it would not be so much fun to detect something or recognize something very special and new or novel that you can identify. You've mentioned you're having a bias against small molecules, yeah? How did you experience?
Well, based on what I've been talking about for the last are you surprised?
No. So this shaped your views on the challenges?
But that's only half of it. Small molecules are anything but specific. And we're seeing this revolution in pharmacology and in treatment modalities such that very specific, highly selective treatments are now available in the form of injections, antibodies and proteins that are highly specific. And I think I'm just comparing that level of specificity to small molecules, which tend to be much less specific.
Was it maybe due to the fact that to that time it was easier to go for small molecules and the technology doesn't exist in this broadness to go for antibodies, proteins, and things like that? That the people prefer to go, or the companies prefer to go to small molecules?
That has been the case through my career. I don't know if that's still the case now. I haven't been in a company for a while. Boy, there sure are a lot of new therapeutic modalities coming out that are...
So in hindsight, when you're looking at this project or other project, is there anything you would have done differently now with the experience you have in the development or handover process of the project or any project? You remember?
Anything I would have done differently? I can't say. I'd have to think about that.
It's not easy. Sometimes I'm also asked this question. I say, and my preformed answer is always, to that time, it was to my best experience.
That's what's in my head. We made decisions based on the data at hand. All we can do, right?
Yeah.
We can always second guess after we have results, but that's not fair.
That's not fair now. And
No.
you would have been the one that you are if you not have gained this experience from what you have learned so far, I would say.
I think one thing I've learned is to be more forceful in not accepting every target that a therapeutic area might think is important, because a lot of targets just are not druggable, as you well know.
The story of this drug's development highlights how science often progresses. It does it in unexpected ways. What advice would you give researchers that are working on projects that have unclear applications?
Oh boy, think out of the box. Make as many collaborations with every type of interesting but unexpected possible hints as to where to go with it. I think a lot of that's going to come from academics. I don't think we have that expertise typically in pharmaceutical companies. But academics, I think, I wouldn't be surprised if the NK3 receptor now is involved in female healthcare. Where did that come from? It mitigates what do you call it, night sweats and female postmenopausal or menopausal symptoms. The NK3 receptor, how did they find that? I'm sure there's many other examples of that, but they have a target and they thought they knew what it would be good for, but it ends up being good for something else.
More generally speaking, or thinking as someone like you with decades of experience, and what advice would you give to young researchers in drug discovery or especially in life sciences?
Have fun. Yeah, have fun.
Yeah, that's right.
It's all about that. If you're really interested in this and you're doing, it's fun. Wonderful.
Mike, thank you. This was fantastic. Thank you so much for sharing your story and these insights with us. I'm convinced that also the audience will really appreciate it that you shared this with us today.
I appreciate being recognized. It's been a long road. It's fun to see some recognition.
Wonderful.
And it's good to see you too. It's been a while.
It's been a while. It's been a long time, yeah. Mike, thank you.